the hydrothermal vent ecosystem. The submersible Alvin dropped through 2,500 meters of absolute darkness in February of 1977, and the scientists inside were expecting nothing. The deep seafloor was supposed to be a desert, cold, lightless, nearly lifeless. Every textbook said so. Every expedition before this one had confirmed it. Geologist Jack Corliss and oceanographer John Edmond were down there to map hydrothermal vent fields on the Galápagos Rift, hot water seeps predicted by plate tectonics. The geology was expected. What clustered around it was not. Corliss reportedly radioed the surface with something close to disbelief. Isn't the deep sea supposed to be like a desert? Well, there's all these animals down here. The discovery was published in Science in 1979, and biologist J. Frederick Grassel described it as potentially the most significant biological find of the 20th century. He was not overstating the case, because what Corliss and Edmond had found didn't just surprise biology, it forced biology to rewrite its most fundamental rule. Every food web known to science ran on sunlight, captured by plants, passed upward through animals, all the way from a blade of grass to a lion. The assumption wasn't a hypothesis. It was the foundation, and the creatures living around those vents didn't use it at all. They ran on chemistry. Sulfur compounds seeping from fractures in the Earth's crust converted to energy by bacteria through a process called chemosynthesis. No sunlight, no photosynthesis. Life that had never needed the star at the center of our solar system, and apparently never would. The tube worms were the first shock. Riftia pachyptila grows up to 2.4 meters long and has no mouth, no stomach, and no digestive system whatsoever. Its entire nutrition comes from chemosynthetic bacteria living inside its own tissues, a symbiotic arrangement with no parallel in any biology observed above the vent zone. The water itself at the black smoker chimneys reaches 400 degrees Celsius without boiling, held liquid by pressures of approximately 250 atmospheres at depth. The Pompeii worm, Alvinella pompeiana, lives at the chimney's edge, tolerating temperatures up to 80 degrees Celsius, the highest sustained temperature tolerance confirmed in any complex animal. In 1985, researchers discovered the blind shrimp Rimicaris exoculata at mid-Atlantic ridge vents, an animal that had sealed over its conventional eyes and grown a light-sensitive organ on its back to detect the faint infrared glow of the vent fluid itself. Vision reinvented not for light, but for heat. Across more than 700 species identified at hydrothermal vents globally, approximately 90% exist nowhere else on Earth. Giant white clams, Calyptogena magnifica, grow up to 26 centimeters at the Galápagos Rift vents, 10 times the size of their shallow water relatives, and live up to 100 years, sustained entirely by internal bacteria rather than anything they filter from the water. In a 2016 review published in the journal Astrobiology, NASA identified hydrothermal vent ecosystems as the primary model for potential life on Europa and Enceladus, moons of Jupiter and Saturn believed to harbor subsurface liquid oceans with active hydrothermal systems. And at the base of every vent food web sit the chemosynthetic bacteria themselves, members of the domain Archaea, the oldest known branch of life on Earth, dating to approximately 3.5 billion years ago, and possibly the closest living relatives of the very first organisms that ever existed. The mainstream explanation holds together. Chemosynthesis is simply another energy pathway. The same logic as photosynthesis applied to chemistry rather than light. Life exploits available energy wherever it finds it. Strange, certainly, but not miraculous. Hydrothermal vents are a niche, and biology fills niches. But if vent life is just another niche, why does 90% of it exist nowhere else on Earth, not adapted to the vent, originating there? And why do the oldest organisms at the base of this ecosystem belong to the oldest known branch of life on the planet? Geochemist Michael Russell and biologist William Martin have published work arguing that life didn't begin at the surface ocean at all, that it began at hydrothermal vents in the chemical heat of early Earth's seafloor billions of years before photosynthesis was invented. If that's true, the surface world, sunlight, plants, animals, everything we think of as life, isn't the story. It's a branch. And the original story is supposedly still running down there, unchanged for 3.5 billion years, on sulfur and heat and chemistry, waiting in the dark. The question that follows from that is one science hasn't fully answered. If life began without sunlight here, what does that mean for where else in the universe life might already be running? In subsurface oceans, next to the heat, waiting for someone to descend far enough to find it? From life that rewrote the rules of its own existence to life that has apparently solved the problem of dying altogether. The immortal jellyfish. Every cell accumulates damage. Every organism, given enough time, fails. This is not a rule of biology in the way that gravity is a rule of physics. It is deeper than that. It is the definition of what it means to be alive. And somewhere in the Mediterranean Sea, a jellyfish roughly the size of your smallest fingernail has apparently not received this information. Turritopsis doherni was first collected in the Mediterranean in 1883 and classified as entirely unremarkable. It sat in the literature for over a century, unexamined and unconsidered. In 1996, German marine biologist Christian Sommer was studying the species at a laboratory in Genoa when something happened that shouldn't have been possible. A mature specimen, rather than dying when injured and starved, began reverting. Its bell contracted, its tentacles withdrew. Its adult cells converted into a different type of cell entirely. It became a polyp, the juvenile form it had started as months earlier, and began its life cycle again from the beginning. The paper documenting this appeared in Invertebrate Biology in 1996, received almost no attention, and sat quietly in the scientific literature for years. Shin Kubota of Kyoto University eventually dedicated his career to the species. Under his observation, individual specimens completed the reversion process more than 10 times in laboratory conditions, each cycle documented, each return to juvenility confirmed. The mechanism is called transdifferentiation, the conversion of a fully specialized adult cell into a different type of specialized cell. In almost all known biology, differentiated cells are permanent. A muscle cell stays a muscle cell. A nerve cell stays a nerve cell. Turritopsis doherni performs transdifferentiation at the scale of an entire organism, converting every adult structure back to its juvenile cellular state. This is not slowing aging. It is not pausing it. It is running the clock in reverse. Kubota's laboratory work, described in detail in Maria Konovalova's 2012 profile in the New York Times, confirmed the repeated cycling. Then, in 2022, researchers at the Universidad de Oviedo published a genomic analysis in PNAS that found something deeper. Turritopsis doherni possesses approximately twice the number of copies of DNA repair genes as its non-immortal close relative Turritopsis rubra, including genes associated with telomere maintenance, the very mechanism whose progressive failure drives aging in most animals. The reversion itself is triggered by environmental stress, starvation, injury, or temperature shock, meaning the organism's immortality is not passive but responsive. It chooses, at the cellular level, to begin again. And the comparison to human biology is not merely metaphorical. The technique of partially reprogramming mammalian cells into a more juvenile state, developed by Shinya Yamanaka and awarded the Nobel Prize in Physiology or Medicine in 2012, is the closest human science has come to replicating what this jellyfish performs naturally. That work remains partial. The jellyfish does it completely, in minutes, repeatedly. Maria Pia Milieta of Texas, A&M University documented in a 2009 paper in the Journal of the Marine Biological Association that the species has already spread to every major ocean, almost certainly transported in ship ballast water, meaning a potentially biologically immortal organism is currently establishing permanent populations in ecosystems it never evolved for worldwide. The mainstream explanation is reasonable. Biological immortality in simple organisms isn't entirely without precedent. Some tardigrades and planarian flatworms show related mechanisms. Turritopsis dohrnii evolved a robust transdifferentiation response, likely as an adaptation to the unpredictable conditions of the Mediterranean's upper water column. Its immortality is real, but constrained. It can still be eaten, killed by disease, or destroyed by environmental disruption. In the wild, most individuals presumably die of external causes long before completing a single reversion cycle. But the 2022 genome study shows the species invested twice the genetic machinery in DNA repair and telomere protection as a closely related species that doesn't revert. Evolution doesn't maintain expensive systems without sustained selection pressure. Something has been selecting for this continuously for an unknown period of time, and the question that follows isn't purely biological, it's almost philosophical. The ocean has been running a working solution to cellular aging in silence at a size barely visible to the naked eye, while human science has spent decades and billions of dollars attempting to replicate the same mechanism artificially. What else down there solved problems we haven't thought to ask about yet? From a creature that dissolves the boundary between life and death, to one that dissolves the boundary between individual and collective in the open ocean at depths where almost nothing else dares to go. The siphonophore. Somewhere in the mesopelagic zone, between 200 and 1,000 meters below the surface, something drifts that is simultaneously the longest animal on Earth and not technically an animal at all, depending on which definition of animal you decide to use and whether you're prepared for that definition to fail you. The Schmidt Ocean Institute filmed it in 2020 off the coast of Western Australia. A specimen of Praia dubia measuring approximately 47 meters in length. A blue whale, the largest confirmed individual animal on Earth, reaches 33.5 meters. This exceeded it by nearly a third. Steve Haddock of the Monterey Bay Aquarium Research Institute, who has produced the most extensive behavioral documentation of siphonophores beginning in the 1990s, would not have been surprised. He had been watching these creatures long enough to understand what they are, which is to say, something that biology's existing vocabulary handles with difficulty. A siphonophore is a colony. But that word is not adequate. Each unit of the body is a zooid, a genetically identical clone that has specialized so completely for a single function that it cannot survive independently. Nectophores jet-propel the colony through the water. Dactylozoids carry the stinging tentacles used for prey capture. Gonozoids handle reproduction. Pneumatophores form the gas-filled float at the top. None of them perform any other function. None of them can. They are not parts of an animal. They are animals that have surrendered their individuality so completely that they have become parts. The entire colony begins from a single fertilized egg that clones itself repeatedly into these differentiated forms. Evolutionary biologist David Queller describes it as one of the clearest examples of a superorganism in nature. Genetically a single individual, functionally a distributed society. The hunting strategy of Nanomyakara, a smaller but better studied siphonophore, was documented by MBARI researcher Kelly Sutherland in a 2017 paper in Nature Communications. The colony spreads its tentacles in a glowing curtain up to 3 meters wide, attracting prey with bioluminescence, then contracts to trap whatever has approached. No central nervous system coordinates any part of this process. And in a 2021 paper published in PNAS, Joanna Dabiri and colleagues analyzed the pulsing sequences of nectophore firing in siphonophore colonies and found the coordination produces hydrodynamic efficiency exceeding that of any known swimming organism. A colony with no brain, composed of thousands of independent organisms, optimizes its own fluid dynamics better than anything that evolved a nervous system to do the same job. The deepest confirmed siphonophore colonies have been recorded at approximately 8,000 meters. A 2017 paper in Deep Sea Research estimated that deep-sea siphonophores collectively consume more fish annually than any other predator group in the ocean, though this remains debated given the difficulty of measuring populations at depth. And the Portuguese man-o-war, that iconic surface-floating danger, is itself a siphonophore. Its gas-filled sail is a single zooid that has become the colony's float. It cannot be removed without killing everything else, yet it is technically one individual organism that has become a body part. Biologists will tell you, fairly, that colonial organisms evolved incrementally, beginning as clusters of identical cells, progressively specializing into differentiated types. Siphonophores represent an extreme end of a continuum that includes corals and bryozoans. Whether they are one organism or many is largely semantic. Evolutionary theory predicts that sufficiently integrated colonies will eventually be classified as individuals regardless of how the question is phrased. But the hydrodynamic efficiency result is not semantic. It is a measurement. A committee with no executive, no coordination mechanism, no brain of any kind, outperforms every individual organism that evolved a nervous system specifically for the purpose of coordinating movement. How does that happen, and what does it imply? Some researchers frame the siphonophore as a thought experiment made biological. What happens when the unit of selection stops being the individual? Praia dubia may be the ocean's clearest answer. And if that answer unsettles the way you think about individuality, consider that every cell in your own body made the same surrender a very long time ago so that you could exist to be unsettled by it. From a creature assembled across evolutionary time, to one that has been suspended in it, unchanged, for 125 million years in the submarine canyons off Japan. The goblin shark. The jaw fires before you see the shark move. Held in place by elastic ligaments rather than rigid bone, it launches forward out of the skull at 3.14 meters per second, covers approximately 1/3 the length of the shark's head, and retracts, the entire strike completing in 322 milliseconds. A human blink takes approximately 400 milliseconds. By the time your eye closes, the jaw has already returned. This is slingshot feeding, and it exists in no other vertebrate on Earth. Mitsukurina ostoni was first described scientifically in 1898 from a specimen caught off Japan, named partly for U.S. Fish Commissioner Austin. Fewer than 50 specimens have been properly documented in more than 125 years of scientific record. The jaw projection mechanism wasn't captured on video until 2008, when researcher Carl Moore filmed a goblin shark feeding at approximately 1,340 meters depth in the Gulf of Mexico, incidentally, not by design. The footage was analyzed by Kazuhiro Nakaya of Hokkaido University, whose 2016 paper in the Journal of Zoology provided the first detailed biomechanical analysis of the strike. For over a century before that footage, the mechanism existed only in anatomy. No one had seen it work. The elongated pink rostrum, the distinctive blade-like snout, is covered in electroreceptive pores called ampullae of Lorenzini at a density higher than any other shark species. It functions as a biological metal detector, sensing the electrical fields generated by prey hidden in sediment. The jaw fires toward what the snout detects, not what the eyes see. And the jaw itself is hydraulic, simultaneously projecting forward and generating suction toward prey, a dual mechanism trap that compensates for the goblin shark's slow overall swimming speed. The translucent pink color of the skin, unusual in any shark, is incidental. The blood vessels sit close enough to the surface to show through, a structural consequence of living at depths where skin pigmentation provides no evolutionary benefit and apparently no selection pressure ever addressed it. The goblin shark's lineage traces to approximately 125 million years ago, making it structurally contemporaneous with Spinosaurus. It belongs to the family Mitsukurinidae, and it is that family's only surviving member. The oldest fossil assigned to the lineage, Scapinorhynchus, dates to approximately 100 million years ago. The morphological similarity was so strong that the 1898 specimen was initially thought to be a living Scapinorhynchus. DNA analysis places the species' last common ancestor with modern sharks at 80 to 95 million years ago. The lineage has been genetically isolated since the Cretaceous. Everything else in its family is extinct. The mainstream explanation is coherent. Deep-sea environments are physically and thermally stable. Over geological time, organisms adapted to them experience reduced selection pressure to change. Living fossils survive because their niche doesn't disappear and their design remains functional. There's no evolutionary incentive to alter a working system. But deep-sea stability is not a shield against mass extinction events. The Cretaceous-Paleogene extinction affected deep-sea species significantly and eliminated every other member of the Mitsukurinidae alongside large portions of the marine fauna. 3 mass extinctions came and went. The goblin shark's ballistic jaw, trident snout, and pink translucent skin survived all of them, apparently unchanged. The question isn't simply how. It's why this specific mechanism, out of everything that existed alongside it, was the one that came through. Some researchers suggest the goblin shark isn't best understood as a survivor, but as a record. Evidence that the slingshot jaw, developed in the Cretaceous, represents an answer to deep-sea ambush predation that 125 million years of subsequent evolution has not improved upon. Every other predator that evolved in that interval found a different solution. None of them built a ballistic jaw. Whether that says something about the jaw's perfection or about the limits of what evolution manages to find in 125 million years of trying, we may never know. From a predator unchanged since the dinosaurs, to one whose survival strategy involved becoming something that defies classification entirely in the lightless, oxygen-depleted zones of the tropical Pacific, the vampire squid. At 600 meters depth, the oxygen level drops to roughly 3% of what exists at the surface. Almost nothing can breathe there. Almost nothing chooses to. Vampirotuthis infernalis, the vampire squid from hell, as its Latin name translates directly, didn't arrive at the oxygen minimum zone by accident. According to the evidence, it evolved to exploit the one zone of the ocean where active predators cannot follow it, then gave up hunting altogether. German biologist Carl Chun first described the species in 1903 from specimens collected during the Valdivia expedition. The animal was placed immediately in its own order, Vampiromorphida, neither squid nor octopus, but a lineage that diverged before the squid-octopus split approximately 300 million years ago. It is not a strange squid, it is a surviving branch of the cephalopod tree that predates both groups. For more than a century after its discovery, what it actually ate remained genuinely unknown. In 2012, Henk Jan Hoving and Bruce Robison of the Monterey Bay Aquarium Research Institute published a landmark paper in Proceedings of the Royal Society B that answered the question, and the answer was stranger than the animal itself. Vampiroteuthis infernalis does not hunt, it harvests marine snow. Marine snow is the ocean's continuous drift of dead organic matter. Dead plankton, fecal pellets, shed skin, degraded particles, falling perpetually from the surface world toward the seafloor below. It is, in essence, the ocean's waste, and Vampiroteuthis infernalis evolved to eat it. Two long retractile filaments extend up to 8 times the body length, potentially 2.4 meters on a 30-centimeter animal, and are covered in tiny hair-like structures maximizing surface area for particle capture. The filaments are unique to this species and have no structural analog in any other known cephalopod. The Hoving and Robison paper analyzed stomach contents and mucus composition across 108 specimens. Marine snow particles accounted for the entire diet, making this the only non-predatory cephalopod known to science. The oxygen minimum zone at 600 to 900 meters contains approximately 0.5 milliliters of oxygen per liter of water, compared to roughly 7 milliliters per liter at the surface. Vampiroteuthis infernalis possesses hemocyanin, the copper-based protein that carries oxygen in its blood, with the highest known oxygen affinity of any cephalopod, allowing it to function at concentrations that would suffocate virtually all other active animals. Its eyes, up to 2.5 centimeters in diameter on a 30-centimeter body, give it the largest eye-to-body ratio of any animal alive. And rather than an ink sac, useless in an environment with no ambient light and no visual predators capable of entering the zone, it possesses a bioluminescent fluid sac. When threatened, it releases a cloud of glowing blue mucus, disorienting predators with light instead of darkness. This is a direct inversion of the defensive mechanism of every other cephalopod that has ever evolved. Genetic analysis places the divergence of Vampiromorphida at approximately 300 million years ago. Only one species survives in the entire order. The niche logic is sound. The oxygen minimum zone provides refuge from predation. Marine snow provides a continuous, passive food source requiring almost no energy to obtain. Every adaptation follows from the constraints. Biologists are right to frame this as elegant exploitation of an available niche. But a 300-million-year-old lineage, living in a predator-free zone with unlimited passive food, should have diversified into multiple species over that span. Vampiroteuthis infernalis has been the sole member of its order for 300 million years. What happened to everything else that shared that lineage? And what does it mean that only this one form, in this one impossible zone, survived whatever eliminated the rest? Some researchers have proposed that what we observe in this animal may not be the original form of the creature at all, that it may be a reduced form compressed over millions of years into the single viable strategy its environment permits. It found the one place it could still exist, gave up hunting, gave up ink, gave up activity, did progressively less until it found the minimum viable version of being alive. Whether that is a triumph of adaptation or something closer to retreat is a question the oxygen minimum zone cannot answer. From the Pacific's smallest predatory mystery to the Southern Ocean's largest invertebrate, an animal we have captured exactly once in over a century of scientific inquiry. The colossal squid, we have found one adult ever. We know it exists primarily because sperm whales carry its beaks in their stomachs. Beaks larger than any squid ever brought to the surface alive. Something moves through the Southern Ocean at depth large enough to leave 5-centimeter hook scars on 11-meter whales, large enough to be measured in fractions of a ton, and invisible to every camera science has ever lowered into the water to find it. Mesonychoteuthis hamiltoni was named in 1925 from 2 arms recovered from a sperm whale's stomach. For decades, the species existed as fragments, isolated beaks in whale digestive tracts, occasional tentacles in deep trawl nets. The first juvenile was captured in 1981. In February of 2007, New Zealand fishermen hauling a Patagonian toothfish longline in the Ross Sea at approximately 1,600 meters depth brought up a living adult. It weighed 495 kilograms. It measured 4.2 meters in total length, with a mantle alone of 2.5 meters. It was frozen and transported to the Museum of New Zealand Te Papa Tongarewa, where a team led by Steve O'Shea of Auckland University of Technology examined it in 2008, broadcast live on New Zealand television. That examination remains the most complete scientific observation of the species ever conducted. No complete adult has been captured since. The eyes of the 2007 specimen measured 27 centimeters in diameter with a 9-centimeter lens, the largest confirmed eyes of any animal, living or extinct. Eric Warrant and Dan Erik Nilsson of Lund University modeled the optical function of eyes this size in a 2012 paper in Current Biology and found them optimized specifically for detecting the bioluminescent glow produced by approaching sperm whales at depths of 120 or more meters. The eyes may not have evolved primarily to find prey. They may have evolved to see the predator coming. The rotating hooks embedded in cartilage rings along the arms, swiveling, tri-pointed barbed, create an escalating grip that tightens in response to struggling prey, requiring greater force to escape with each attempt. Circular scars on sperm whale skin matching colossal squid hook dimensions at up to 5 centimeters in diameter confirm that these animals resist capture and inflict significant injury on the whales before being subdued. The ammoniacal flesh of Mesonychoteuthis hamiltoni, saturated with ammonium chloride providing neutral buoyancy at depth, renders the meat inedible and foul-tasting, which is likely why the species has never been commercially fished. Biomass estimates from deep-sea research and related literature suggest populations in the millions of tons in the Southern Ocean, though these figures are poorly constrained by any direct observation. The species belongs to the family Cronchidae, the glass squids, and retains the transparent body plan of that family at enormous scale, potentially rendering it visually undetectable at depth despite its size. Cold Southern Ocean water supports gigantism in cephalopods through slower metabolic rates and longer growth periods. The colossal squid's rarity in collections reflects the genuine difficulty of deep-sea sampling rather than actual scarcity. At the biomass levels whale stomach contents imply, it is probably common at depth. These are fair explanations, and biologists who offer them are not wrong. But if the colossal squid constitutes a major component of sperm whale diet, eaten daily by animals we track with satellite tags and acoustic monitors, why has every attempt to film one in its natural environment failed? We have hours of footage of sperm whales diving to hunt at depth, we have never captured what they are hunting. Some deep-sea optics researchers have noted that the Lund University eye modeling implies the squid evolved to detect precisely the light signatures that ROV cameras and research submersibles emit. If the animal can see the lights of every vehicle we've sent to find it before that vehicle comes into range, our failure to film it is not a sampling problem. It is a visibility problem. The sperm whales find them daily. What are the whales doing that, after 100 years of trying, we are not? From the largest eye evolution has ever constructed, to a fish that built something smaller, stranger, and in some ways more sophisticated, inside a skull made of glass. The barrel-eye fish. For 65 years, scientists described this animal incorrectly. Every specimen collected had a flat-topped head with two eyes pointing straight upward, an odd but catalogued configuration, filed and moved past. It wasn't until 2004, when Monterey Bay Aquarium Research Institute researchers sent an ROV with low-light cameras to depth, that anyone saw what the fish actually looked like while it was alive. The flat-topped head was an artifact of collection. The transparent fluid-filled dome covering the entire upper skull had been collapsing under pressure change every time a trawl net reached the surface. A century of description had been studying a collapsed version of something it never understood. Macropinna microstoma was first described in 1939. The footage from 2004, published in 2009 by Bruce Robison and Kim Reisenbichler in Coppea, showed the dome fully intact for the first time, a fluid-filled transparent shield spanning the entire dorsal head within which two bright green cylindrical eyes rotated freely. Not fixed, rotating. Pointing straight up to scan for prey silhouettes against the faint light filtering down from the surface, then rotating forward to direct captured prey toward the mouth. The same muscles that pivot the eyes also reshape the tubular lens, adjusting focal length for near versus far targets as the eyes move between scanning and targeting positions. The optical system is tubular rather than spherical. The same principle as a telephoto lens compared to a wide angle, maximizing light gathering capacity at the near total darkness of the mesopelagic zone. The visual pigments in those eyes are maximally sensitive to wavelengths around 500 nanometers. A green pigment layer inside the eye filters shorter wavelength bioluminescent emissions, roughly 480 nanometers, produced by other deep-sea organisms while passing downwelling sunlight at 500 to 520 nanometers. The fish can distinguish the shadow of a prey animal above from a bioluminescent flash below. It filters the world by wavelength automatically, without computation, using pigment chemistry. The dome itself is composed of fibrous connective tissue filled with fluid, structurally equivalent to a corneal dome but scaled to cover the entire skull. The fish has, in effect, externalized its optics to head scale, making the dome an optical element rather than merely a protective cover. MBRE researchers documented the species apparently stealing food from the trapped tentacles of siphonophores, implying the green spectral filter allows it to operate near bioluminescent structures without being overwhelmed by them. The species reaches approximately 15 centimeters in length, with a small, upward-pointing mouth positioned directly below the upward-pointing eyes. The entire geometry of the head is designed for detecting and acquiring prey from above with no wasted structure. Biologists explain the tubular eye as a well-documented mesopelagic adaptation. Multiple unrelated species evolved the same configuration independently for the same reason: maximum light collection at depth. The dome evolved to protect mobile eyes from the stinging cells of the siphonophores the fish feeds near. The rotation mechanism elegantly resolves having upward-scanning eyes and a forward-facing mouth. All of it follows from the niche. But the dome isn't only protective. Its refractive properties contribute to visual acuity in ways a purely transparent shield would not provide. The fish didn't evolve a cover for its eyes. It evolved its skull into an optical instrument. And marine optics researchers have noted, with something between admiration and unease, that the resulting system, tubular geometry, spectral filtering, wide aperture dome, mechanical rotation with focal adjustment, independently replicates the core design principles of astronomical telescopes built from biological tissue, operating at light levels 1/1000th of what exists at the surface. Two very different processes, evolution and engineering, apparently converged on the same solution to the same optical problem. Whether that parallel means something or is simply a case of optimal solutions being limited in number regardless of the path taken to find them is a question that sits uncomfortably between disciplines. What is genuinely strange is not that evolution built this. It is that a century of science studied the broken version and didn't know what it was missing. From a creature whose design was invisible for 65 years, to one that has been visible and largely unchanged for 80 million years in the deep Atlantic slopes off Japan. The frilled shark. It looks like an eel. It moves like a snake. It has 300 teeth arranged in 25 rows, each tooth a three-pronged trident whose inward-facing cusps drive deeper into anything that attempts to pull free. And it has been carrying its young supposedly for 3.5 years, longer than any other vertebrate on Earth, through water that was already ancient when the dinosaurs were alive. American ichthyologist Samuel Garman described Chlamydosalacus anguineus in 1884 from specimens caught off Japan. He recognized immediately that he was looking at something outside the standard classification. The 6-paired gill slits, each fringed with a collar of frilled tissue, matched the configuration of Paleozoic sharks from 300 million years ago. Standard sharks have 5. Garman placed it in its own family on the spot. DNA analysis published across multiple studies places the species' last common ancestor with modern sharks at approximately 80 to 95 million years ago. The lineage has been genetically isolated since the Cretaceous. The oldest fossil, assigned to its family, Cladosalach, dates to approximately 380 million years ago, and the frilled shark retained that ancestor's gill configuration while every other shark lineage eventually moved to 5. The jaw is terminal, at the front of the head rather than underneath, as in nearly all modern sharks. Another ancestral configuration that allows forward strikes at fast-moving prey without the need to pass them below the body first. The highly flexible vertebral column, far more mobile than in rigid-bodied modern sharks, has led some researchers to propose that the species may coil and strike in a manner similar to a snake, a behavior that would explain how it captures the fast cephalopods found in its stomach contents, though this has been proposed in the behavioral literature and never directly filmed. Stomach analyses from captured specimens document bony fish, cephalopods, and other sharks. The 300-tooth trap works whether the prey struggles or not. The inward-facing cusps ensure that withdrawal drives the points deeper at every attempt. The gestation period of approximately 3.5 years was estimated by Shotonica and colleagues from reproductive tract analysis across multiple specimens, correlating embryo development stage with water temperature. At 14 degrees Celsius, the standard temperature of the species' depth range, 3.5 years is the result. This exceeds the 22-month gestation of an elephant. A female alive today may be carrying pups conceived in 2021. The embryos develop without a placenta, sustained entirely by a yolk sac throughout the process, meaning the female invests continuous metabolic resources for 42 months while also hunting at depth. The species has been encountered alive fewer than a dozen times. The 2007 footage from Awashima Marine Park in Japan, filmed shortly before the captured specimen died, remains among the most complete visual records available. The standard explanation is that deep-sea environments are thermally and physically stable over geological timescales. Organisms adapted to them evolve slowly. The frilled shark's niche didn't disappear. Its design remained functional. The long gestation reflects metabolic constraints of a cold, low-energy environment. Living fossils exist because stability, not disruption, is the dominant condition of the deep. But deep-sea stability is not a firewall against mass extinction. The Cretaceous-Paleogene event affected deep-sea species significantly. 3 mass extinction events came and went across the frilled shark's lineage. Every modern shark family that shared the Cretaceous ocean with it is extinct or transformed. The 6 gill slits, the trident teeth, the terminal jaw, none of them changed. The question isn't just how the design survived, it's why the deep ocean, which supposedly changes slowly, preserved this one so exactly while eliminating virtually everything around it. Some researchers frame the frilled shark not as a survivor, but as evidence that the deep sea runs on a different temporal logic. The pressures that drive rapid evolution on the surface, competition, climate disruption, ecological cascades, may operate so slowly at depth that 80 million years looks from the ocean floor like an unremarkable interval. If that's true, the frilled shark isn't a relic. It's a demonstration that certain answers once found can simply persist, unchanged, unimproved, sufficient, for timescales that make evolutionary history difficult to grasp. From a predator frozen in Cretaceous design to an animal that inherited a design from before the age of dinosaurs and then scaled it to something that demands a different kind of explanation in the Gulf of Mexico, the giant isopod. You know this animal. It rolls into a ball when disturbed. It lives under rocks and leaf litter. You've probably held one in your hand or watched a child hold one because it's harmless and abundant and entirely familiar. A roly-poly, a pill bug, a woodlouse. Now put it in the deep ocean and wait 160 million years. Alphonse Milne Edwards first described Bathynomus giganteus in 1879 from a specimen trawled in the Gulf of Mexico at approximately 400 meters. The relationship to the common pill bug was immediately unmistakable and immediately puzzling. The same armored body plan, the same defensive role, the same compound eye arrangement, the same multi-directional jaw system. The difference was scale. Where the terrestrial Armadillidium vulgar reaches 2 centimeters, the deep-sea version reaches 76. The largest confirmed specimen was captured in the Gulf of Mexico in 2010 and measured 76 centimeters in length. Preliminary phylogenetic analyses show approximately 98% genome similarity between the 2 species, meaning 160 million years of divergent evolution and a 38-fold increase in body size, with the same biological blueprint underneath both forms throughout. The Toba Aquarium in Japan documented the starvation record. A specimen arrived in January of 2009. Aquarium staff offered food on multiple occasions across the following years. The animal refused every offering. February 9, 2014, 5 years and 43 days after its arrival, it died. The cause of death was not identified as starvation. It is the longest confirmed starvation survival record for any crustacean and one of the longest for any animal of comparable size. The 4 sets of jaws, maxillulae, maxillae, mandibles, and maxillipeds can operate in concert or independently, processing prey in 4 simultaneous cutting directions. The compound eyes contain a large number of facets maximizing light-gathering surface area, fixed rather than mobile, but providing a wide visual field at near zero light levels. Stomach content analyses document whale falls, fish carcasses, and slow-moving living prey, including sea cucumbers and sponges. Feast or famine feeding that matches the abyssal plain's episodic food supply. A new species, Bathynomus raxasa, was described from Indonesian waters in 2018, suggesting the genus is substantially more diverse than the approximately 20 known species indicate. And the defensive ball posture, locking calcified dorsal plates together under significant mechanical force, is retained fully in Bathynomus giganteus despite the apparent absence of any predator at its operating depth whose attack the behavior could plausibly deter. The mainstream explanation applies established deep-sea biology. Cold water slows metabolic rates, allowing larger body size to develop over extended growth periods. Reduced predation pressure means large body size is survivable. Feast or famine food availability on the abyssal plain favors organisms that can store energy and endure long intervals between meals. These are real mechanisms. Biologists invoking them are not reaching. But the Toba aquarium specimen complicates the metabolic framing. That animal was held in warm, well-oxygenated water, conditions that should have elevated its resting metabolism and increased the cost of fasting. Food was presented repeatedly and declined repeatedly. A metabolic explanation accounts for the capacity to endure starvation. It does not account for the choice. The deeper strangeness of Bathynomus giganteus may be precisely its familiarity. It takes a body plan that has worked for 160 million years across every terrestrial environment on Earth. A design so successful it has never been replaced in the habitats it occupies, and scales it until every parameter transforms. The eyes that protected against birds now gather light at 2,000 meters from sources that have no name. The jaws that processed leaf litter now process whalebone. The defensive ball that closed against a sparrow now closes against whatever moves in the abyssal dark. The deep ocean didn't invent this animal, it inherited a working blueprint, removed most of the external constraints, and has been exploring how large the design can go before something gives way. At 76 centimeters and 5 years without food, apparently, nothing has given way yet, from the animal that refuses to die without food. Back to the floor of the oldest living system on Earth, where a shrimp sealed its own eyes and grew new ones on its back to see a light that has no business existing. The eyeless eye. It has no eyes, not in the sense that some deep-sea animals have reduced vestigial eyes still present but non-functional. The sockets where eyes should be are fused over with tissue. A deliberate closure made somewhere in evolutionary history, when the calculus of maintaining visual organs in total darkness stopped making sense. But it can see, not with its face, with its back. And what it sees in a place with no light is light that has no right to be there. Rimicaris exoculata was described in 1986 from specimens collected at the Tag hydrothermal vent field on the Mid-Atlantic Ridge, at depths between 2,300 and 3,500 meters. The sealed eye sockets were noted and filed, unremarkable in the context of deep-sea biology. What came next was not unremarkable. In 1989, Cindy Van Dover, then a graduate student at Woods Hole Oceanographic Institution, later the first woman to pilot the submersible Alvin solo, noticed a reflective patch on the dorsal carapace of live specimens while reviewing ROV footage. The patch was a photoreceptor organ. It had no lens, no cornea, no focusing apparatus of any kind. It was a flat, naked sheet of light-sensitive cells lying directly beneath the transparent carapace. The minimum viable photoreceptor, stripped of every structural component except the light-sensitive cell itself. The discovery appeared in Nature in 1989. The function was confirmed in 2000 by Elke Nobel and colleagues in the Journal of Experimental Biology. In situ measurements at the TAG vent field by Van Dover and colleagues confirmed the emission of near-infrared photons in the 600 to 750 nanometer range from superheated vent fluid. This wavelength range overlaps precisely with the spectral sensitivity of the dorsal eye organ. The organ detects the thermal radiation of the vent, not reflected light, but heat glow. The shrimp swarm around vent chimneys in densities of up to 2,500 individuals per square meter, feeding on chemosynthetic bacterial mats growing on vent surfaces. The dorsal organ allows them to maintain close proximity to their food source without direct contact with vent fluid that reaches 350 to 400 degrees Celsius. They navigate the boundary between survivable and lethal temperatures using something that functions as vision. The Nobel 2000 paper confirmed that the organ's photoreceptors are rhabdomeric cells, the same cell type used in conventional crustacean compound eyes, arranged in a flat sheet beneath the carapace rather than the curved array of a compound eye, and with no structural mechanism for forming images. It counts photons, it detects direction, it answers one question. The shrimp simultaneously carries the sealed vestigial sockets of the eyes it abandoned and the functional dorsal organ it grew to replace them. Two evolutionary experiments with vision running in the same organism, one closed and one open, representing a biological record of evolution changing its approach entirely. Molecular clock analysis places the species' colonization of the mid-Atlantic ridge at approximately 6 million years ago. Both transitions, the closure of conventional eyes and the development of the dorsal organ, occurred within that window, though which came first has not been resolved. The mainstream explanation is straightforward and accurate as far as it goes. This is regressive evolution followed by de novo sensory adaptation. Eyes were lost because they were costly and provided no benefit in total darkness. The dorsal organ evolved because the thermal radiation of vent fluid created a specific survival-relevant selection pressure that a photoreceptor could address. Every step follows from the environment. The organ is stripped down because stripping down was sufficient. But the transition happened twice simultaneously in the same animal, running in opposite directions, eyes closing as the back opened. And the new organ was built not from new biology, but from the same rhabdomeric cells the abandoned eyes had used, now reorganized into the simplest possible configuration. Rimicaris exoculata didn't invent vision. It reinvented it from recycled parts for a different light in the last place on Earth where you would expect anyone to need eyes. The dorsal organ of this shrimp may be what vision actually is at its irreducible minimum. Not an image, not a scene, not a face, but the answer to a single question. Is the lethal thing closer or farther away? Every visual system that has ever evolved, from the compound eye of a trilobite to the fovea of a human, is ultimately an elaboration on that one question. This animal stripped every elaboration away and kept only the question. And in the 2016 Astrobiology review that cited the 1977 Alvin discovery as its primary model for extraterrestrial life, NASA noted that whatever lives in the subsurface oceans of Europa or Enceladus would likely be doing something similar, surviving in the dark next to the heat on chemistry rather than light. If something sees down there, it may have arrived at the same solution. built for one question: in the dark, beside the heat, on a world where the sun never mattered and never will. Pull back. From the vents and the transparent skulls and the ballistic jaws and the naked retinas navigating heat in the dark. Look at what remains when you set the details aside. 10 species, every ocean basin, geological timescales ranging from 6 million years to 3.5 billion, and in every case, the same event. A rule that biology considered settled was broken at the point of discovery. Life requires sunlight. Organisms age and die. Animals have a single body. Ancient lineages disappear. Eyes need lenses. These weren't hypotheses requiring confirmation. They were the foundations of the discipline, and the deep ocean, one by one, quietly broke each of them. The mainstream view is coherent, and it deserves to be stated fairly. Chemosynthesis is an alternative energy pathway. Transdifferentiation is a real mechanism with real evolutionary logic behind it. Colonial organisms exist on a continuum. Living fossils survive in stable niches. Deep-sea gigantism follows from cold water metabolics. None of the scientists who built these explanations were wrong to build them, and none of the explanations are false. They are accurate as far as they reach. What they don't account for is the accumulation. 10 items, 10 different fundamental assumptions, each failing in a different way, in different ocean basins, across different time scales. At some point, a list of exceptions stops feeling like exceptions and starts feeling like a correction. Not to the science, but to the assumption underneath the science that we already understood the boundaries of what life is permitted to do. There is a philosophical dimension here that sits outside the data. The deep ocean is older than almost everything we think of as history. It was running its own experiments in the dark, at its own pace, by its own logic, before the first tree grew on land. When we finally descended into it with instruments capable of observing it properly, we found that it had already solved problems we didn't know were problems. Cellular immortality, collective intelligence without a nervous system, vision built from heat, life without a star. Supposedly, these are anomalies. Some researchers believe they are something closer to a reminder that the version of biology we built by studying the surface world is a partial map, and the ocean has been holding the rest of it for a very long time. We have mapped roughly 20% of the ocean floor. In that 20%, we found all of this. The other 80% has not been examined. That is not a caveat. That is an open question of extraordinary scale, and the only honest response to it is to keep descending. You made it here. That means you're already the kind of person who does. Hit subscribe. The descent continues. And go to the comments with this. Which of these 10 species do you think has the largest implications for what we think we know? Not the strangest looking. The one whose existence, if fully understood, would change the most. Build the case. The ocean floor was here before the continents took their current shape, before the first eye opened toward sunlight, before anything we call history began. It ran its own experiments in the dark, on its own timeline, and by its own rules. Rules that turned out to be different from the ones we assumed it shared with us. In February of 1977, a submersible dropped through 2,500 meters of what science had confidently declared lifeless and found the original story still running. We have seen 20% of it. The other 80% doesn't care whether we're ready.